Hello friends. Welcome to the Tech Science YouTube channel. I know the best updating make you success. So I try to give best informations that you should know every time. Today we talk about tests that can use for identifying the COVID-19 patients. Before that, I request you to subscribe my YouTube channel and give support. Okay, let's move to the video. Firstly, we will see how coronavirus was spreading from the beginning to now. You know, what's the test name to detect coronavirus? Okay, I will tell you. Actually, this strain of the coronavirus COVID-19 does not have a readily available test kit, but it can nevertheless be detected by a series of tests in the hospital. So do you know which labs can conduct the coronavirus tests? Tests for the virus can only be conducted in specialized labs that are certified to do so. What actually happens during a coronavirus test? Let me explain you step by step. Firstly, the lab will acquire one of the following samples from the patient. First, a swab test. The lab will take a special cotton swab and sample the inside of the throat or nose. Second, a nasal aspirate. The lab will inject a saline solution into your nose, then remove the sample with gentle suction. Third, a tracheal aspirate. A thin, lighted tube called a bronchoscope goes into your lungs, where a sample will be collected. Fourth, a sputum test. Sputum is a variation of mucus from your lungs that can coughed out or sampled from the nose with a swab. Fifth, a blood test. The collected sample will be analyzed for the virus, either through a blanket test for all variants of the coronavirus including regular flu, or through a specialized gene sequencing test that locates the marker for the novel coronavirus. The next question is, when should you take a test for coronavirus? The symptoms of COVID-19 include fever, cough, shortness of breath, sore throat. However, patients may only need testing if you have symptoms of infection and have recently traveled to parts of the world where infection rates are high. You may also need testing if you have had close contact with someone who has traveled to one of those areas. I think you have already heard the name PCR test. Let me explain about polymerase chain reaction. Polymerase chain reaction, PCR, is a method used widely in molecular biology to make millions to billions of copies of a specific DNA sample rapidly, allowing scientists to take a very small sample of DNA and amplify it to a large enough amount to study in detail. PCR was invented in 1983 by the American biochemist Kary Mullis. It is fundamental to much of genetic testing, including analysis of ancient samples of DNA and identification of infectious agents. Using PCR, copies of very small amounts of DNA sequences are exponentially amplified in a series or cycles of temperature changes. PCR is now a common and often indispensable technique used in medical laboratory and clinical laboratory research for a broad variety of applications, including biomedical research and criminal forensics. PCR tests work by detecting specific genetic material within the virus. The throat swab is well suited for polymerase chain reaction testing, while the blood sample will be mined for antibodies specific to the new disease, known as COVID-19. Both tests take just hours to run. meaning results could be ready within a day. However, once the rapid form of these PCR and antibody tests are created, results could be ready in under an hour. Once a sample arrives at the lab, researchers extract its nucleic acid, which holds the virus genome. Then, researchers can amplify certain regions of the genome by using a technique known as reverse transcription polymerase chain reaction. This, in effect, gives researchers a large sample that they can then compare to the new coronavirus, known as SARS-CoV-2. SARS-CoV-2 has nearly 30,000 nucleotides, the building blocks that make up DNA and RNA. The PCR test developed by the University of Washington School of Medicine, UW Medicine, targets just 100 nucleotides that are specific to SARS-CoV-2. These 100 nucleotides include 2 genes in the SARS-CoV-2 genome. A sample is considered positive if the test finds both genes, inconclusive if just one gene is found, and negative if neither gene is detected. In contrast, serological tests look for specific antibodies that the body has produced to fight the virus. If they detect those antibodies, the test gives a positive result. A serological test is advantageous because it can detect antibodies even if a patient has recovered, whereas a PCR test can detect the virus only if the person is currently sick. However, both tests might miss cases if samples are taken too early, when the viral load is too low, or if the person's body hasn't produced antibodies against the virus yet. For the serologic test, it can take about a week before the body produces ample antibodies, although as we get more serologic tests, there will be guidance on what time frames are needed to do a serologic test. However, nowadays scientists have found so many ways to identify the symptoms fastly. So guys, stay in your home and give the best support to them for recovering this situation. So guys, I hope you all got some knowledge about tests of COVID-19 virus. Thank you for watching this video, and please stay with my channel to get some new science and technology news.